Good day, everyone. I am Nikiwe Mhlanga from Mintech, and I'll be giving a brief talk on the AMD Mintech CHPC cluster, focusing mostly on the surface enhanced Raman spectroscopy project, which is the development of CES biosensors for infectious diseases. I am going to introduce my institution, its research, and the different research groups. So AMD is a research and development institution that focuses on mineral processing and metallurgical engineering products and services to industries worldwide. It has three technical clusters which carry the load of the research and development, and they are the mineral processing and characterization cluster, which is subdivided to mineralogy, analytical chemistry, and mineral processing. Secondly, we have the extraction metallurgy cluster, which is which houses the metallurgy divisions, which is your biometallurgy, your hydro, and your pyrometallurgy. And the third, the last but not the least, is the mining, materials, and automation cluster, which houses measurements and control, advanced materials, and the mining and mineral economics. And I am from the advanced material division, which is also abbreviated as AMD, is mandated to serve the national interest through research, development, and technology transfer. That is to promote mineral technology and foster the establishment and expansion of industries in the field of minerals and products which will be derived from that. Slide 5 sort of summarizes the AMD division. It houses four research groups, which is the catalysis, physical metallurgy, health platform, and nanotechnology. The use of the HPC platform started with the catalysis group, which they were mainly using it for the HIESA project, which deals with the development of fuel cells. It later then expanded into the physical metallurgy and the health platform group. The physical metallurgy mostly uses it for calculations to enhance their production of memory-shaped alloys. And in the health platform, it is used by the diagnostic group. The diagnostic group focuses on the fabrication of diagnostic kits or diagnostics for infectious diseases such as malaria, HIV and AIDS, and the recent COVID-19. So as I said before, this talk will focus mostly on the diagnostic group and with a focus on the surface enhanced Raman spectroscopy biosensors. So the CES phenomenon has been around for the last 4 to 5 decades. And in a nutshell, it's just basically the amplification or the enhancement of what could have been a very weak traditional Raman signal in the presence of surfaces or substrates such as roughened plasmonic metallic nanoparticles, that is your gold, your silver, and your copper nanoparticles. And the substrates have even expanded to your two-dimensional materials such as graphene. So the sketch over here shown in figure 1 shows the architecture of a traditional CES biosensor whereby you have a support, and then on the support you are going to chemically assemble the CES substrate in the form of metallic nanoparticles. It could be gold or silver, and then on that you will chemically bind a capture antibody. The purpose of the capture antibody is to identify and capture the antigen of interest. So if maybe this was a malaria biosensor, this would be a malaria antigen. So it binds it at a certain epitope. And then in addition to that, you are going to have a detection antibody which also identifies and binds the antigen, but at a different epitope compared to this one, so there is no overlap. This one also, which is the detection antibody, is also conjugated to the self-substrate in the form of the metallic nanoparticles. In a case whereby what you analyte, what you're trying to sense is Raman active, it does have characteristic Raman fingerprints, then you don't need the self-substrate ends here, you don't need the Raman reporter. However, mostly biological molecules are not Raman active. So in that case, then we need a Raman reporter or a Raman tag. And thiols are most popular purpose of this is to indirectly confirm the presence of this via the characteristic peaks of the Raman tag. And it confirms that indirectly because everything here is chemically conjugated. So there is no way you can actually have this in the absence of that. I hope that is clear. So our division adopted the CES platform because of various reasons such as high sensitivity and the fact that you can both qualitate on quantity, like it's both qualitative and a quantitative detection method, has a capability of multiplexing, and it is a single molecule fingerprint. To come up with a SaaS platform which is reproducible and is very much rich in most of those properties which I've just mentioned. We sort of need to understand the chemistry of even the precursor materials before you can put them together. You sort of need an insight into their chemistry, their conjugations, and the HPC platform has enabled that for us. For example, mostly we use citrate-capped nanoparticles in the synthesis or in the fabrication of our system. So we needed to understand how the citrate ion binds or sits on the metallic clusters. And we were able to use Monte Carlo to actually get an idea of how actually it binds and what even happens as you increase the amount of the citrate ion onto the clusters, as it's shown by these images by figure 2 over here. By figure 2 over here, it seems like the citrate ions, they actually prefer a certain facet of the nanoparticles, and as you add more, they actually pack on top of the others in that particular site which they prefer. And this here sort of informs the experiment in the sense of how much of the citrate you're supposed to use in your experiment. And in addition to that, it also controls the size because everything is size dependent as well. So most mentioned that our system, since we're sensing biological molecules, so it needs to have a Raman tag. In our case, we are using 4-mercaptobenzoic acid as our Raman tag, and it is known to have the acid end on one side and the thiol end. So using the HPC platform, we're also able to study the chemistry of its binding into the nanoparticles. What exactly does it favor? Which facet does it favor? Which end favors which facet, So we're able to do a study on that, and we're able to deduce that there's even differences between the nanoparticles themselves. What maybe silver favors is not what gold favors in terms of the facets and the binding terminals. And then since the whole platform is going to be using biological molecules, we're also able, using Strondiger and Monte Carlo, to actually introduce a biological molecule whereby we downloaded Plasmodium falciparum antibody from the protein bank, and we're able to see how exactly to study its interaction with the clusters which have been conjugated to the tag, and we're able to deduce that it's actually a physisorption reaction. It's not based on the differences on the distance between the two atoms, or like the two entities. So all of this facilitated the whole production or the whole synthesis of the biosensor in the lab, whereby we went into the lab and were able to synthesize the biosensor, and then we tested it even on malaria infected blood, non-infectious, of course, supplied by WHO. From the blood was from different regions, which is Nigeria, Santa Lucia, and the Philippines. What's worth noting is also the fact that the blood had different parasite loadings in it, and our system was able to actually track that, was able to capture the fact that it had, so to sort of quantify the different loadings of the parasite in the different bloods. And also interestingly, we did a repetitive study to find out how fast it can actually capture, and we're able to go as low as 3 minutes. Our system was able to show like Raman peaks, 4 MBA peaks, even at 3 minutes. Of course, with more time, then there was more enhancement. So on the on the malaria CES biosensor, we were able to do that. Then beginning of, is it beginning of, it was late 2019 to beginning of 2020, we had the COVID pandemic sort of sweeping through our world. And then as a diagnostic group, we also sort of try to jump into that to try and see what we can come up with in terms of diagnostic. We had a group which was working on rapid diagnostic kits, the antigen and the antigen and antibody tracking ones, and I was mostly on the group for the SES, where we try to adapt the existing SES platform or probe for COVID. The main idea was to try and use the ACE2 COVID receptor as our detection, because it will be able to identify the COVID antigen. We had one antibody at that time. We only had one COVID antibody, which we used as our capture. So what we did, we also used the HPC platform, Strondasia, to after that, we downloaded a SARS-CoV-2 spike receptor from the protein bank, and then we were able to analyze it and identify reactive amino groups, which were then later of the ACE2, which we then synthesized in the lab and we conjugated with our CES tag and silver or gold. And this was tested on an antigen, a COVID antigen. However, it gave very poor sensitivity. So in a nutshell, those are the two projects that we've been able to use the HPC platform on. So it's mostly the study of the material, the chemistry of the material that we use in our different products as a diagnostic group. So the second part of the presentation, which is talk a bit on why we need access to the HPC resources, the challenges and research impacts. So we surely need access to these various codes because we don't have a license to them. So we're really grateful to CHPC for access to the codes. And then we also run very large systems. And for us to be able to run our systems at CHPC is really great because our computers cannot be able to handle most of the systems that we need to run, and the calculations also, the deep chemistry calculations that we need to run, even studying your orbitals and etc. So all of this, it facilitates the laboratory experiment, and once we get that right, then we can be able to have prototypes. So there is, I think the impacts thus far is that we are able to produce papers from this, and we've had students graduating as well, and then we have more students in the pipeline, and then we also were able to register a prototype, the malaria cells prototype, and we at stage of cleaning it up, and we hope it can go through to clinical trials and pass that and be able to be used by the medical department. Thank you so much to CHPC for access to these different codes. It wouldn't be